everyone. So we are finally at the last topic of this particular chapter and we'll be finishing it, no? Okay. So starting with this one, in this particular lesson, we'll be learning about the different kind of animal tissues, continuation from the previous slide. And the new topics that we have here are the muscular tissue, nervous tissues, and the synapses and the working of the muscles. Okay, working of muscles is not as fundamentally explained in the NCRT, but it will help you in the longer run. So we are going to cover that one. Okay, now moving to revising, first of all, what actually is the animal tissue. Okay, animal tissues are the one things which can vary means which could be different from each other from the structure point of view, function point of view, and the origin point of view. Origin means they could be originating from different layers of the embryo. Okay, we'll be learning more about that in the higher classes, and they're divided into epithelial, connective, muscular, and nervous. Epithelial is done, connective is done, and we'll be doing the muscular and nervous tissue in this particular lesson. Okay. Mm. So moving forward to what actually are the muscular tissue, we'll go by the key points again because key points are going to save us in the exams. Okay. If you don't remember anything, but you just remember the key points about a certain topic or a certain organ or a certain tissue, you can write a story about them. Okay. Like, right. So moving forward, muscles are basically the soft tissue. Okay. Secondly, they give rise the body they give rise the ability to contract to the body. Remember when I told you about how the muscles have the ability of causing the movements in our body with the mechanism of contraction and relaxation? That is what we'll be talking about here as well. Okay, and in the mammals, since we are talking about the human beings and human beings are the mammals, and mammals are the organisms which give rise to the young ones and feed them with their milk. Okay, they have the mammary glands basically. So the mammals, they basically have four types of muscles. Three type of muscles. We have the cardiac muscles, we have the smooth or the skeletal muscles or the striated muscles, and we have the smooth or the non-striated muscles. Okay, we'll be covering each of them one by one, and we'll be seeing how exactly each of them is very, very important and wherever this is present according to the function and the structure. Okay, now see, first of all, we'll be discussing about how exactly there is difference in the shape of all these three muscles, okay? And then we'll be moving forward to how do they function or wherever these are present, okay? So moving to this and giving you guys the diagram, we have this right here, okay? All right. So what do you see here? You see how exactly we have the cardiac muscles and how cardiac muscles, they are attached with each other. Okay, and the attachment points right here and right here, these attachment points are called as the intercalated discs. Okay, and these intercalated discs make sure that these muscles are attached together and these muscles are communicating with each other, and they are working in a rhythmic manner and in a synchronized manner, in a collective manner altogether, right? Have you ever seen the heart beating separately one by one, the different part of the hearts beating separately, like first part beating and then second part beating? No, right? The entire heart beats it like lub lub dub lub dub, right? That is because of these intercalated discs. Okay, then we have the striations. Do you see the different kind of light and dark bands which are present in these muscles? These are the ones which we call as the striations. These are present because of the actin and myosin filaments. We'll be talking more about these. Okay, and then we have the skeletal muscles. Skeletal muscles are the ones which also have these striations. That is also because of the actin and myosin filaments. The major thing is that skeletal muscles, they are the voluntary muscles, or they basically are the control they are under the control of the human mind. For example, my hands, I'm moving my hands, I'm moving my lips, I'm dancing right now. That is because of the skeletal muscles. Okay, next we have is the smooth muscles, which are not under our control at all. For example, the muscles of our lungs, because we are breathing. Okay, the lungs are expanding, they are exchanging the air, right, the gases, oxygen and carbon dioxide, but we do not have any control over that. Or for example, the stomach and the intestine, we just eat food and the stomach and intestine digest the food and absorb the food. Again, they do it in their own time, we do not have any control over that. And they do not have these striations as well. Okay, these do not have, oh my bad, these do not have any striations. Okay, striations as in they do not have any actin or myosin filaments like that. They just have one single kind of filament. Okay, and what is that one single kind of filament? We'll be talking about that more. Okay. So basically, these smooth muscles are the spindle shaped muscles. Okay, spindle shaped doesn't they have tapering ends like this, and they're like fat in the middle and they have tapering ends like that. Okay, so that is one thing. And now you have a fair idea of how these muscles are like, how these look like. We'll be now talking about how these have different kind of components and how do they function. Okay. So starting with these cardiac muscles, first of all, I'll be start writing right here. We have the cardiac muscles. These cardiac muscles are the ones which are responsible for the beating of the heart, and there is only one in one particular place in the body where these cardiac muscles are found. These cardiac muscles are found in the heart. Okay, cardiac related to heart. Okay, and where are these found? In the heart. So these are basically found in the heart, and these are the muscles which are anti-fatigue. When I say anti-fatigue, that means they are untiring muscles. They are never going to get tired in your entire life. They just start beating from from your birth, from when you start forming as a baby inside the mother's mother's tummy, and till your death. Okay, so your heart just keeps on beating and beating and beating, and just trying to circulate and pump the blood into the entire body. And they have the rhythmic contraction and relaxation, with the help of which the heartbeat is generated properly. Okay, so I'm gonna write they're found in the heart. These are untiring muscles. Untiring muscles. These generate heartbeat. Okay, they have intercalated discs, which we have already discussed about. Okay, I'm pretty sure you must have noticed, you must have noted that down. And they have actin and myosin. They have actin and myosin filaments, right? And these are branched, have branches. As you can already see in the particular diagram, these have different branches protruding out like from one another. Okay, and that is why the entire heart is like connected together and stuff. And they have multiple nucleus. Have multiple nucleus. Okay, that is one thing about the cardiac muscles. Next we have is the skeletal muscles, or we can also call them the voluntary muscles, okay, or the striated muscles. Why the striated muscles? Because they have the striations, they have the alternating light and dark band. Okay, and what exactly is that used for? We'll be talking about that in the upcoming slides. Okay, So, these skeletal muscles are the ones which are under our direct control, and they have a long cylindrical shape, okay, and they basically are parallel to each other, as I already showed you in the diagram. And these muscles, they have the light and dark bands of the actin filaments and the myosin filaments, okay. Myosin filaments are the dark bands and actin filaments are the light bands. Okay. So have dark band, which is made up of the myosin, okay, which is very, very thick as well. And they have the light band, which basically is the actin filament, and it is very, very thin. Okay, just make sure that you notice that they are under a direct control, and we'll be talking more about these when we'll be covering the functioning of muscles, okay, or the mechanism of muscles functioning or working. Next we have the smooth muscles. Smooth muscles are the ones which are also called as the involuntary muscles, okay, or they're also called as the non-striated muscles or unstriated muscle, one and the same thing. Okay, why are they called so? Because they do not have any alternate striations. They are just they do not have a proper, pattern of arrangement of actin and myosin filaments. Okay, they just have the actin filaments which are attached to one another, and they're very, very dense, right? Apart from that, they have a spindle shaped body which is tapering at both ends and fat in the middle, and they, since our involuntary muscles are present in the organs which do not have which do not are under control, like direct control, okay, which do not are which not are under direct control, right? So that is one thing about the smooth muscles, okay. And they have this single nucleus, okay. And filaments, muscle filaments is what I'm talking about, are present in a homogeneous mixture. Now, by the homogeneous mixture, I mean that they are mixed with each other so that there is no alternate dark and light band. Okay. That being said, we'll move forward to the working of the muscles. And in the working of the muscles, we'll be talking about how the skeletal muscles function and have the proper mechanism of working. Okay. Now, see, when we are talking about the working of the muscle, remember when I told you how we have the two alternate bands, we have the dark band, which is the myosin and which is very, very thick, and we have the light band, which is the actin and which is very, very thin. They have a proper arrangement. Okay, so we'll go by the key points first of all, and then we'll move forward further. So muscular tissues, they have the elongated cells or they have the cylindrical cells, which we have already talked about, okay, which are called as the muscle fibers. The actin and the myosin fibers make up the actin and myosin filaments make up the muscle fibers, okay, and it is responsible for the movement of our body, right? Also, main function is to provide the movement again. Now see, we have these special proteins in our muscles which are called as the contractile proteins, and these contractile proteins are the ones which do the entire movement, okay. So we'll do one thing. I'll tell you about the different kind of bands which are present inside the muscle tissue. That will help you in understanding of how exactly these work. Okay. So we have the light band, we have the dark band. We also call them as the sarcomere and stuff. I'll be talking about that. See, first things first, these are attached with one another in a very, very synchronized, in a very, very folded manner. Okay, these are folded individual units, and then all individual units are packed together, and those units are then packed together. And if we kind of separate each unit, we'll see how different fibers are attached, okay, or how different fibers are arranged with each other. So I'm going to do one thing. I'm going to show you that with the help of a particular diagram right here. Okay, so what I have for you here is this particular diagram, and what you see in this one is you see how everything is attached with one another, okay, and how it is packed very nicely. So you see this is one particular filament of the skeletal muscle. You can see the nucleus, you can see the light and dark band right here alternating. You can see the muscle fiber. One muscle fiber is what we're talking about, which will have multiple mitochondria in order to produce the energy, and will have the sarcolemma. What is a sarcolemma, bacha? Sarcolemma is something which forms the tubules in the entire muscle fiber, and it is present in the proper intervals, and it helps in the movement of the muscle. Okay. So think of sarcolemma as a component which is forming the tubules, tube-like structures in the skeletal muscles, in the skeletal muscle fibers, and it is present at the regular intervals so that it can help in the proper movement of the body, okay, proper movement of the muscle. And one single unit is called as the myofibril. One single unit is called as the what? Myofibril. Now, what will happen if we take out and enlarge this one single unit? We'll see how there is sarcoplasmic reticulum. Sarcoplasmic reticulum is nothing but the smooth endoplasmic reticulum, which you must have learnt about in the previous chapter. And the smooth endoplasmic reticulum is the one which will have a lot of calcium reservoir, and that is why it is called as the sarcoplasmic reticulum. Why it does need to have a lot of calcium reservoir? Because those calcium ions are the ones which help in the proper movement of the muscles. Okay, now you see we have the arrangement of the thick filaments and the thin filaments. Thick are the myosin and thin are the actin filaments. Okay, now see, these thick and thin filaments are going to have a dark band, which is which basically is the myosin filament, light band, which is the actin filament. Okay, then there is a zone which is called as the H zone. Okay, H zone is the one which will just have the myosin part in the entire muscle. Okay, and it is present right here. This H zone will just have the myosin filaments. Okay, then it also has the Z disc. Now see, Z disc is something which is uh, let's say lengthwise. When every fibril has multiple sarcomeres, sarcomere is the one working unit of the muscle fiber. So these will be differentiated with the help of the zigzag lines, which are called as the Z lines. Okay, so starting from one Z line to the another Z line, we will have one working unit of the muscle fiber. Okay, so this is like the one working unit of the muscle fiber right here, starting from one Z disc to the another Z disc. This is the Z disc which is present. Okay, this is the Z disc that we are talking about. I'll draw that right here with the help of the black color. And what you see right here, this is the Z disc or the Z line which we're talking about. And starting from one Z disc will move to the another Z disc, and this is called as the sarcomere. And what is sarcomere? Sarcomere is the one working unit of the entire muscle. Okay, one working unit of entire of the entire muscle. Now, see, important part about this is that these sarcomeres make the entire um, uh, muscle fiber, and these different units come up and collectively make one particular muscle fiber, which helps in the proper functioning, right? Now we'll do one thing. We will be learning about how there's a cross bridge connection formation and how there's the proper working of the muscles through this particular video. And if there's any need be, just let me know. If I'm your teacher in the live classes, or just let your other teacher know, and I'm sure they'll perfectly handle it. Okay. So starting with this, and I'll be explaining everything in between as well. Okay.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomere.
Remember when I told you about the sarcomere, how they start from one Z line to the another Z line, right? And they have an H band and multiple bands together, which make one functional unit. Moving forward.
Each sarcomere contains many parallel, overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released.
Remember when I told you about the smooth endoplasmic reticulum, which has a lot of calcium reservoir, and now we're talking about the movement. So this is the movement. Okay, this is the movement. And when I'm moving my hand, my muscles are contracting, and when I'm relaxing my hand, my muscles are also uncontracting or relaxing. Okay, moving forward, we just have the filaments sliding over each other like this, and that is how the muscles contract, and we just relax. So when we relax, muscles kind of slide off each other, the filaments slide off each other. Okay, and everything is stimulated whenever there is the calcium flow into the body. Okay, moving forward.
The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding site.
See, the troponin and the tropomyosin that we're talking about right here are the proteins which are present alongside the muscle fibers. And whenever the calcium occurs, whenever the calcium is released, the troponin and tropomyosin are acting, which is resulting into the movement of the muscle fibers. Okay, so I'm going to back it up a bit, and then we are going to study it again in order for better understanding. So now that you know what troponin and tropomyosin are, okay, pay attention, please.
Are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction.
Remember here that this particular view that you're seeing right now is the view of a muscle fiber under a microscope. The muscle fiber, the chain, the purple colored chain that you're seeing right here is the actin filament, and beneath the red colored filament that you're seeing is the myosin filament. Remember that actin filament will have the troponin and tropomyosin proteins, and whenever the troponin protein is bound with the calcium, it is going to replace the tropomyosin. Okay, and it is basically is going to let's say help in the stimulation of movement of tropomyosin protein as well, and that is going to release, or that is going to expose the myosin filament. Okay, and those myosin filament are having what? ADP is adenosine diphosphate. Adenosine diphosphate when combined with another phosphate forms the ATP, adenosine triphosphate, which is what? Which is the universal energy unit formed by mitochondria. Now moving forward, pay attention.
The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin.
So what this particular person is trying to say here is that when the myosin was exposed, it bound with the actin filament and they moved over one another, causing the movement, causing the contraction. But when the energy came, when the ATP came, it came and bound with the myosin again, and the bridge between the two actin and myosin filaments was hampered. And when it was hampered, the bridge was deformed, the bridge was separated. Okay, moving forward.
The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
So now I hope that you guys understood how exactly things happen. Okay, that is all about the muscle tissues. And now we'll be moving forward to the neurons or the nervous tissue of the human body. Okay, alrighty, moving forward. So now we'll be talking about the nervous tissue. You must have learned about nervous tissue in the previous classes as well. The important thing about the nervous tissue is that it is specialized. It is specialized for what? Communication. And this communication occurs between the brain, between the spinal cord, between the peripheral nerves. Peripheral nerves are the ones which are present in our entire limb, in the abdominal, in the face. Okay, and we basically have two type of nervous tissue. We have the neurons and we have the glial cells. Important thing is, bacha, neurons are the fundamental unit of the nervous system, and we'll be learning more about that, whereas the glial cells are the smaller tissues which are present in the nervous system, and their major function is to maintain the neurons. Okay, which is the actual fundamental cell. Okay, and they also help in what? In the transmission of signal, okay, along with the neuron. So that is what glial cells are there for. And the neurons, if we come back to the neurons, neurons are basically responsible for communicating with each other through electrical and chemical signals. How that actually happens, we'll be moving forward with that. Okay, we'll be learning that as well. So first of all, we'll be learning about the different parts of neurons and different functions which are carried out by the different parts. Okay, so I'm going to give you one single diagram which will solve all the problems. Okay, so I hope everybody can see this particular diagram. We'll be talking about the different kind of parts of neuron. First of all, this is the neuron cell. Okay, and this will have to learn, and this is very very important for the exam point of view. Okay, a lot of diagram for neurons are asked in the exam, and this could be a 3-mark question or a 5-mark question. Now back to the parts. We have the dendrites. First of all, the tentacle type things that you're seeing in the neuron are the dendrites. Dendrites are the ones which are responsible for receiving the signal from the other neurons. Okay, and these neurons are arranged end to end like this. Okay, these are arranged end to end like this, and they're making a complete trail. And these dendrites, they help in receiving the signals from the other cells, from the other neurons. Then we have the cell body. Cell body is the one which is which basically has a lot of cell organelles, mitochondria, and the cytoplasm, nucleus, endoplasmic reticulum. It just basically is a regular cell type space. Okay, and then we have the nucleus, which is the brain of the cell, and it helps in controlling everything. Then we have the cell membrane. Cell membrane is the one thing which is present in all the animal cells, and it is responsible for protecting the entire cell. Okay, it helps in also maintaining the structural integrity or the shape of the cell. Then then we have the axon hillock. Now remember this thing, when we are talking about the neuron, we just discussed how these go ahead with passing the chemical and the electrical signals. The electrical signal is conducted through this particular length, okay? This is called as the axon, which is mentioned right here, and chemical signal is conducted through this particular length, which is called as the axon terminal. Okay, we'll be learning about this, how exactly this happens. Okay, moving back to the axon hillock. So axon hillock, whenever the signal is or the impulse is taken up by one particular neuron, axon hillock, this particular part is the part where it is converted into electrical signal from the chemical signal. Now when it is connected, when it is converted from is chemical to electrical signal, then it is going to pass through the entire axon length. Okay, important thing about the axon length is that this axon length will have the Schwann cells, okay, and the myelin sheath. The blue colored, light blue colored sheath that you're seeing right here, or the light blue colored component that you see right here, is called as the myelin sheath, and this myelin sheath is responsible for presenting in the entire axon length, and it increases the speed of the signal. How does it increase the speed of the signal? Because then signal will have to jump like this. Okay, it won't have to travel the entire length. Why does it have to jump? Because myelin sheath is insulated. Myelin sheath does not conduct the electricity. Okay, so only small spaces are left, which we call as the nodes of Renvir, through which the signal is jumping like this, and that jumping is making it pretty fast. And when we have the Schwann cells, think of Schwann cells as the glial cells, okay, type of glial cells which are present, which basically help in the production of myelin sheath, maintenance of neuron, and help in the transmission of signal, right? Then we have the axon terminal. Axon terminal is the terminal which basically is responsible for converting the electrical signal back into the chemical signal and transmitting the signal to the other cells. Okay, so this transmits the signal to other cells, right? That being said, we have the synapses. Synapses is one thing, bacha. Synapse is nothing special, but it basically the space between the two consecutive neurons or the two adjacent neurons which are attached or which are arranged in an end-to-end manner or end-to-end fashion. Okay, so synapse is the place between the axon terminal of one neuron and dendrites of the other neuron, and into the synapse the neurotransmitter is released. What is a neurotransmitter? Remember when I told you axon terminal is the one which is converting the electrical signal back into the chemical signal and then releasing the signal towards the another cell. That chemical through which the signal is released from the axon terminal is called as the neurotransmitter. And this neurotransmitter is basically a chemical which is received by the another neuron or the preceding neuron. Okay, and the example is acetylcholine. One example is the acetylcholine. Okay, now, moving forward, since this place is called as the synapse through which the chemicals or neurotransmitters are being passed through in order to pass the impulse or the signal, this part, the axon part, will be called as the presynaptic part, and the axon part of the receiving neuron is called as the postsynaptic. Okay, sending cell presynaptic, receiving cell postsynaptic, and the synapse is the part where the two neurons are kind of interacting with each other. Okay, or where the signal is being transported from one to the another neuron in the form of a chemical or a neurotransmitter. Example, acetylcholine. Again, now what is this action potential? This action potential is nothing special but the movement of electrical signal through axon. Okay, there are multiple steps to the action potential as well, which we'll be learning in the higher classes. So most welcome, I'll teach you even more. Okay, now see, going by the this particular definition, we have synapse, or this is also called as the neuronal junction, okay, helps in the transmission of electric nerve between the two nerve cells through transmitter, through neurotransmitter. Okay, what is a neurotransmitter? It is the chemical which is helping, and the example is the acetylcholine. Okay, this acetylcholine is mainly present in the brain. Okay, and whoso ever doesn't have much acetylcholine, they may suffer from different diseases such as dementia, which is the memory loss, or Alzheimer's, which is the degeneration of the brain. Okay, we'll be learning more about that in the actual live class. Now time for some questions. Okay, name the tissue responsible for the movement of our body. Remember when we were just talking about and we just saw a video about it? The tissue which is responsible for the movement of our body is muscular tissue and the nervous tissue. Why muscular tissue? Because the contraction and relaxation. Why nervous tissue? If there will be no signal, the muscles won't move. If the muscles won't move, there'll be no contraction and relaxation, and hence no movement. Okay, now I have one more question. What does a neuron look like? Didn't I tell you how important the diagram of neuron was? There you go. And then we have the solution. Basically, neuron is the cell, and you draw the diagram right here. There's no need of writing the theory. You can just draw the diagram. Okay, and that's it for today, and I'm so very happy. Keep learning from Ask IITians, and I'll see you in the next chapter. Yes, bye bye, take care, kiddos.